Hi everyone, in this lecture we'll talk about the detection of viral infection. The etiological examination of viral infectious diseases is helpful to guide clinical treatment of patients and provide laboratory basis for controlling the prevalence of viral diseases. This timeline shows the major relevant findings in diagnostic virology from 1892 until 2015. Over the past decades, there have been substantial improvements in the detection methods of viral infections. Accurate sample collection is critical for virus detection. Different types of body fluids or tissues can be taken depending on the site of infection and transmission of the virus. Samples used for viral isolation or detection should be collected from patients at the acute stage of disease to improve the positive rate of detection. Antibiotics should be added to inhibit the growth of bacteria or fungi in the specimen. In addition, the collected specimen should be kept at a low temperature and sent for examination as soon as possible. For serological diagnosis, double serum samples should be collected to facilitate the dynamic observation of serum antibody titer. If the antibody titer in the convalescent phase serum sample is at least 4-fold higher than the titer in the acute phase serum sample, the patient is considered to be infected. The growth of viruses requires living cells because viruses replicate only in living cells, not on cell-free media the way most bacteria can. Viral isolation and culture mainly include animal inoculation, chicken embryo culture, and cell culture. Animal inoculation is the first method of viral isolation, which is not commonly used now. However, the virus that cannot be cultured in chicken embryo and tissue cells can still be isolated and identified by animal inoculation. The advantages of animal inoculation include the following aspects: sensitive to virus, large amount of virus cultivated, and study the pathogenic effects of viruses. Meanwhile, the disadvantages are also obvious. For example, higher cost, not easy to manage, easy to cause environmental pollution, and interference with test results due to the immunity of the animal body. In 1931, chicken embryo was used for virus culture for the first time. According to the tropism of the virus, different viruses can be cultured in different regions of chicken embryo, such as the chorial allantoic membrane, allantoic cavity, amniotic sac, and yolk sac. The advantages of chicken embryo culture are easy to operate, easy to source. Most poultry-derived viruses can multiply in, cultivate a large amount of viruses, and can be used for vaccine production. The disadvantages are that mammalian viruses are less sensitive to chicken embryos, and the cost is higher. At present, chicken embryo culture is mainly used to isolate influenza virus or produce influenza vaccine. Cell culture is the most commonly used method in viral isolation and identification. But not all viruses can be isolated or cultured using this method. There are 3 types of cell culture: primary cell culture, diploid cell culture, and continuous cell culture. Primary cells are normal cells derived from the source animal or human embryo tissues. Primary cells are highly sensitive to viruses, but the source of primary cells is difficult to obtain. They can be used in viral isolation and vaccine production. Diploid cells are monolayer cells that remain twice the number of chromosomes after 50 to 100 passages in vitro. They are used for human virus isolation and vaccine production. Continuous cells are derived from the mutation of tumor cells or diploid cells and can be continuously passaged in vitro, such as HeLa cells. They are widely used in virus isolation. However, they cannot be used for vaccine production due to derivation from cancer cells. When viruses multiply in cell culture, they produce biological effects, for example, cytopathic changes, viral interference, and the production of a hemagglutinin that promote identification of the agent. Virus growth in cell culture frequently produces a characteristic cytopathic effect, CPE, that can provide a presumptive identification. CPE is a change in the appearance of the virus-infected cells. This change can be in such features as size, shape, and the fusion of cells to form multinucleated giant cells. CPE is usually a manifestation of virus-infected cells that are dying or dead. The picture shows the CPE of virus-infected cells. We can see virus-infected cells become round and detached from the bottom of the culture bottle. If the virus does not produce a CPE, its presence can be detected by several other techniques. Hemabsorption refers to attachment of erythrocytes to the surface of virus-infected cells. When virus with hemagglutinin infects the cell, hemagglutinin can appear on the cell membrane, which can bind with the red blood cells of the vertebrate. Therefore, this technique is limited to viruses with a hemagglutinin protein on their envelope, such as mumps and influenza viruses. Some viruses doesn't cause CPE, but it can inhibit the CPE caused by another virus when the two viruses infect the same cell. For example, rubella virus, which does not cause a CPE, can be detected by interference with the formation of a CPE by certain enteroviruses, such as echovirus or Coxsackie virus. The diagnosis of viral infection includes morphological observation, detection of viral components, and serological diagnosis. The morphological examination of the virus includes the observation of the morphology and the size of the virus particles in the samples by electron microscope, and the observation of the inclusion bodies produced by virus infection in the tissues by light microscope. Virus components, such as viral antigen and nucleic acid, can be used as a target for detection. Viral protein antigen can be detected by enzyme immunoassay, immunofluorescence assay, Western blotting, and so on. The principle of ELISA is to fix specific antiviral antibodies for antigen detection or viral antigen for antibody detection on the surface of a solid phase carrier in advance, form an antigen-antibody complex with the sample to be tested, and then add enzyme-labeled antibodies or enzyme-labeled anti-antibodies. A ternary complex is formed. After washing, a substrate is added to develop the color and the catalysis of the enzyme. The color depth is proportional to the amount of antigen or antibody in the test specimen. The level of virus in the specimen can be determined by the absorbance value measured by the microplate reader. The ELISA method has characteristics of strong specificity and high sensitivity, but its throughput is small and the detection time is long. CLIA is a new type of immunoassay technology that combines immune response and chemiluminescence analysis. Chemiluminescence labeling immunoassay is to immobilize antibodies on magnetic particles and directly label antibodies, antigen detection or antigens for antibody detection with chemiluminescent agents, and then react with the test specimen to form a solid phase coating complex. After washing, oxidation decomposes the luminescent agent to make it emit light. The CLIA method is one of the most advanced immunoassay techniques at present. It has the characteristics of high sensitivity, strong specificity, and wide detection range. However, it depends on a specific chemiluminescence instrument, and the detection cost is high. Over the past 2 decades, there have been significant improvements in the molecular methods used to quantify viral nucleic acid in clinical specimens. As for nucleic acid detection, there are several methods, including gene sequencing, nucleic acid hybridization, quantitative PCR, ddPCR, gene chip, LAMP, and so on. The biggest advantage of sequencing technology is high sensitivity and high accuracy. However, due to the high cost of the instrument, it depends on the professional's solution to the sequence reading, and the detection time is long. It's currently not suitable for large-scale detection. Fluorescence quantitative PCR TaqMan method utilizes the polymerization activity of Taq enzyme and 5 to 3 primer exonuclease activity to cleave specific TaqMan probes that are complementary to the target sequence during the chain extension process. Due to its excellent sensitivity and specificity, the fluorescent quantitative PCR method is currently widely used. Droplet digital PCR, ddPCR, uses the dropletization technology to divide the reaction system into tens of thousands of droplets for quantitative PCR amplification. After the reaction is completed, the droplet analyzer is used to read the fluorescent signal of each droplet to achieve absolute quantification of template DNA. Its essence is to turn one test of quantitative PCR into tens of thousands of tests. ddPCR technology has the advantages of absolute quantification and high sensitivity, and its detection limit is much lower than that of fluorescence quantitative PCR, which reduces the risk of false negatives and greatly improves the accuracy of detection. However, ddPCR relies on expensive instruments and has a high cost, so its popularity is not high. Gene chip is a detection technology based on the principle of nucleic acid hybridization. A large number of specific oligonucleotide probes are fixed on a chip carrier, such as a glass slide in the form of a microarray, and the hybridization is detected by hybridizing with fluorescent-labeled sample nucleic acid. Use the presence and strength of the signal to analyze the number of composition of the nucleic acids in the specimen. LAMP is a novel method termed loop-mediated isosomal amplification that amplifies DNA with high specificity, efficiency, and rapidity under isosomal conditions. LAMP is an efficient and accurate DNA isosomal amplification technology that can achieve exponential amplification of DNA at 60 to 65 centigrade, and its amplification efficiency is 10 to 1,000 times that of PCR. In addition, because LAMP has designed 4 to 6 specific primers for the 6 segments on the target DNA chain, the specificity is also very high. The principle of serological diagnosis of virus detection is to detect whether there is corresponding antibody in the serum of patients with known virus antigen. Methods of serological detection include neutralization test, hemagglutination inhibition test, complement fixation test, and specific IgM antibody detection. Neutralization test is the test in which viruses lose their infectivity when neutralized by specific antibodies in cell culture. CPE is observed after inoculating sensitive cells with a series of diluted sera mixed with equal volume of known virus suspension. This method is used to detect slight dynamic change of antibody in patient's serum, but less used in clinical diagnosis. Viruses with hemagglutinin can agglutinate red blood cells of vertebrates, which is called hemagglutination. This phenomenon can be inhibited by the corresponding antibody, which is called hemagglutination inhibition. In this test, the known virus is mixed with the patient's serum, and red blood cells are added to observe whether red blood cells agglutination is inhibited. Complement fixation test is a classic method for demonstrating the presence of antibody in patient's serum. It's based on the principle that antigen-antibody complex fixes the complement. As coupling of complement has no visible effects or changes, it's necessary to use an indicator system consisting of sheep red blood cell and coated with anti-sheep red blood cell antibody. Complement lyses antibody-coated red blood cell. It was used to diagnose infections, particularly with microbes that are not easily detected by culture methods. However, in clinical diagnostic labs, it has been largely superseded by other serological methods. IgM antibody appears early after virus infection in human body, so detection of specific IgM antibody is used to diagnose current viral infection. For example, the presence of IgM antibody to core antigen indicates infection by hepatitis B virus. Okay, that's all for the detection of viral infection. Thank you for watching and see you next time.